Hello everybody, now we are moving on to Module 5. We are skipping Module 4, you are not required to do Module 4. And Module 5 is structure-based evidence. So go to your notebook, scroll down until you find Module 5, structure-based evidence. And the first thing we're going to do is figure out what protein family, PFAM, that our protein of interest belongs to. So basically you open a new tab, click here, open a new tab, and that will take you to the PFAM website. So you click on the PFAM website, that will open a new window, and what you do is you click sequence search, and that will pull up a box. So what do you paste in here? You paste in your protein sequence. So you go back up in your notebook to the amino acid sequence here. So you copy and paste the amino acid sequence in. So let me do that. And hit go. So after that's done, what it should pull up is a page like this. So basically, this page tells you what family this protein belongs to. So it belongs to the COB-N magnesium chelatase family. What's the description of this family? Well, it is a magnesium chelatase. It is a cobalt and magnesium chelatase. What that basically means is that it binds magnesium and cobalt. And these are the domains that actually bind magnesium and cobalt in your protein. So you can see here that amino acids 299 to 666 is one cobalt / magnesium binding domain, and amino acids 735 to 1506 is another cobalt / magnesium binding domain. So let's go back to GENIACT and see what information is required for our annotation. So scrolling down. Okay, so we're on structure-based evidence and we are doing PFAM. So what they want is the PFAM number and PFAM name. So let's go here. And what you need is the PFAM name. So this is basically the PFAM name. And they also want the PFAM number. So let's click on family and see where that takes us. Scroll down. And this should be your PFAM number. So PF is PFAM, and this will give you your PFAM number. So let's go back, and this is your PFAM number. This is your PFAM name. You really don't need the scores and things here, but what you need is the pairwise alignment of your protein with the cobalt / magnesium chelating domain, or the magnesium binding domain. So here, you go here, so in your summary. So how did we get here? We went from this page and we basically clicked on the family, and that took us to this page. The PF designation is PFAM, and this is your PFAM family. So you copy and paste this into your lab notebook. This doesn't have a clan, so we couldn't put in the clan. Now what we want to do is look at the alignment of the domain in our protein. So you click on alignment. Okay, where did that show up? Okay, let me just close some tabs and open the link in a new tab. That technically should show us the alignment. Let me see, where did that go? Okay, so if you click directly on there, and basically you generate the alignment. So the alignment comes up as a text file, so I don't recommend you copy and paste this text file. So let's go back into the PFAM family sequence search. There should be an alignment somewhere. Here we go, show, alignment, show. So this is the alignment that you need. It's not the other alignment. That's the alignment of lots of different proteins. So this is the alignment of your protein. This is your protein, and where these domains are located in your protein. So you basically copy and paste the entire alignment into your GenieAct notebook like this. The next thing they need is the HMM logo. So you go back to, no, we don't want this page. We go back to this page, and from here you go to family, and from the summary you look at the HMM logo. So this is the HMM logo. What this basically tells you is where there are conserved sequences in the cobalt magnesium binding domain. So the bigger the amino acid, the more conserved the sequence. So you just scroll along. You basically download this as a file because you can't take a screenshot of something that big. And then what you do is you basically look for conserved sequences. So not that conserved, not that conserved. So these aren't that conserved because the letters are very small. So you look for a stretch of really big letters, and I think I found my stretch of really big letters. So here. So this is a very highly conserved region because the bigger the letter, the more conserved. The region is more conserved. So you can take a screenshot of your conserved regions and then paste them or upload them into your HMM logo and try and predict the key functional residues, which will basically be the most conserved residues. Okay. Then we move on to PDB. So what did PFAM tell us? PFAM told us what kind of family this protein belonged to. It belonged to a family of proteins which can bind either magnesium or cobalt. So now we know quite a bit about this protein. We know that it's located in the transmembrane region. Some of it is extracellular and some of it is in the cytoplasm. So it's a transmembrane protein. It can bind cobalt, and it has some conserved residues or conserved domains that basically help it in its functioning to bind cobalt. Now, if you want to get a structural overview, you need to go to PDB, which is the protein data bank. Now, this is a bit of a hit or a miss because what you're looking for are cobalt binding proteins. So basically you go to PDB. Let's go to PDB. And cobalt binding proteins. And you hit go. Let's see, and this is an archaea. So you look at it in archaea. So we don't have any in archaea, so we can just look at it in bacteria. So let's look at it in bacteria. So that's not exactly what we're looking for. So let's go back, and we're looking for cobalt binding proteins in archaea. Okay, here it is in archaea. Let's see if we find anything. Thiamine aminopeptidase. Yeah, I don't think this is the correct one. So let's go back, look at bacteria. Okay. Yeah, this takes a bit of trial and error. I'm just going to put in cobalt transporter because this is what the protein does. I'm not sure if I'm going to get a hit. It takes a little while to actually find a structural hit in PDB. So cobalt ion transmembrane transporter, okay. We're just going to stick here. Magnesium transporter, magnesium transporter. So if you look long enough, you'll probably find a cobalt transporter. And I've done this before, and this was basically the cobalt part of the cobalt transporter. If you don't find a hit in PDB, because we are annotating ABC transporters, you can put in the general structure of an ABC transporter from PDB. Now we move on to the next module, which is enzymatic function. What we're going to do here is look at which pathways this protein is involved in. This is also a sort of a hit or miss. So what you do is, let me close some tabs up there. What you do is you copy paste or control click, and this will take you to KEGG. Okay, I copied the wrong thing, I guess. Okay, let me just control click this. Okay, that will take you to the KEGG pathway database. And once again, you have to enter your keywords. So since we're doing cobalt transporters and we know that this protein binds cobalt, it makes kind of sense to assume that it is a cobalt transporter. Let's see, cobalt transport. Let's see what that pulls up. Okay, so it will pull up the entire ABC transporter pathway. and we have cobalt here. So let's see, let's look at the pathway. Okay, it's showing you the reference pathway for ABC transporters. Well, it will in a minute. There you go. And what you need is the cobalt portion of that pathway. If your transporter transports something else which is not here, what you need to do is just copy paste the entire pathway into your annotation, but here you just need to copy paste this portion. So what this is it kind of gives you the structure of a cobalt transporter. The cobalt transporter contains 4 domains: the CBIN, the CBIM, the CBIQ, and the CBIO domain. So this is what it's telling you. So KEGG tells you which pathway your transporter will function in. And in this case, we know that it binds cobalt. Therefore, it also transports cobalt, so our transporter functions in cobalt transport. Okay. We don't need metapsych or EC number, so the only thing to do now is enter our proposed annotation for the gene. So from all the evidence, what we found was it had 2 transmembrane helices. There was an large extracellular loop and small intracellular regions. It transports cobalt, so basically our gene is a cobalt transporter, subunit N. Okay, so because it can't be subunit M. We don't have enough transmembrane helices. It's not subunit Q. It's not subunit O. So our transporter is subunit N of a cobalamin transporter. It's actually a cobalt transporter. That's wrong. So it's a cobalt transporter. Okay, so what we did during genome annotation is, first of all, I'm just scrolling up. This is what your notebook should look like. Remember to save. So the modules that we did were the basic information module, where we got basic information about the gene and the protein. We got the length of the DNA sequence. We got the actual DNA sequence. We got the length of the protein sequence and the actual protein sequence. Then we looked at the sequence-based similarity data to pull up proteins which were similar to our own in order to figure out the function of our protein. And we found that it chelated cobalt, which means it bound cobalt. Then we looked at the cellular localization data, and what we figured out was that it was located, it had transmembrane helices, therefore it was located in the cytoplasmic membrane. We didn't do the ORF module. Then we went to the structure-based evidence module, and we looked at which protein family it belonged to. And it belonged to the, it had basically a cobalt binding domains. Actually, it had a couple of cobalt binding domains. Then we looked at the enzymatic function, and it is an ABC transporter, which is responsible for transporting cobalt. We didn't do the gene duplication or the horizontal gene transfer or RNA modules. You don't really need this to annotate your genes, but if you wanted to look at, say, for instance, evolution of this gene, you could have done the duplication and degradation module to look at gene death and gene birth. and we didn't do the RNA because these are very new genes and there aren't really any non-coding RNA that have been documented for these genes. So basically we did the basic information module, the basic information module, which gave us some basic information about a gene and protein. We did the sequence-based similarity data to figure out which proteins were similar to our own, because similar proteins usually have a similar function. Then we looked at cellular localization, and we found that this protein was localized to the plasma membrane. Then we looked at structure-based evidence and found that it had cobalt binding domains. We looked at KEGG pathways and found that it was an ABC transporter, which probably transports cobalt. So using all this information, the final step is annotation or naming of the gene. So let me change this here. No, I've already saved it, so I can't change it. It's actually a cobalt transporter because it transports cobalt, and we have the CBIN gene. So I named this as the cobalt transporter subunit N. So prokaryotic genes are actually arranged in operons. So 1, 2, 3, 4 operons. One operon with the 4 genes makes or codes for this transporter. And our particular gene is the CBIN, and this is how it's arranged in the genome. So it's CBIM, CBIN, CBIQ, and CBIO. And we basically annotated the CBIN gene of the cobalt transporter, and that completes our genome annotation. Of course, you will all have different genes. Most of you will have transporters, but there are a few of you who don't have transporters, because basically we just ran out of transporters in the genome that we are annotating. So now we're annotating Methanobacterium species strain SWAN1, and it apparently doesn't have that many ABC transporters. You annotated a lot of them last fall, and apparently we only have 3 or 4 left, so some of you may have different proteins which may not be transporters. And with that, we end this series of webcasts on genome annotation.